So before talking about oncogenic viruses, of course, we need to look upon the problem and to explain what is actually oncogenesis and what is oncogene. So oncogenesis, it is a process when normal healthy cell of host body, human or animal, can be transformed to cancer cell. Usually that transformation is due to some genetic changes in material of cell due to mutation or other problems. When oncogene is activated or hyperactivated or control over oncogene is lost, and previously normal cell will divide in uncontrolled manner. So first question is, what is oncogene? Activation of oncogene, control over oncogene. So in common words, oncogene is a gene with potential to induce normal cell to cancer cell. So the gene is usually required to control speed and number of multiplication cells. We of course all know, and you should remember from biology, that any cell have known and certain duration of life and certain number of multiplications. And in our body, different cells have different options for multiplication. Some neural cells are non-divided, immune cells are not divided cells, but for example, the same stem cell have almost unlimited ability to multiply. And of course, those differences, they are initially coded by gene which is known as oncogene. So of course the gene is not required to induce cancer, but when control over the gene will be lost or some mutation occurred in the gene, then number of multiplication which is coded by the gene can be lost and cell will lose control over multiplication. So usually, according to different studies in tumor cells, oncogenes are always mutated or expressed at high level. Of course, we have function gene and control genes. So the main function of oncogene is multiplication of the cell. And of course, we have control genes which can express or suppress the gene. If it is required, cells can control higher or in lower rate. And those processes of transformation of normal cell to cancer cell can be due to changes in oncogene itself or changes in other genes which are controlling oncogenes. So nowadays, more than 30 genes identified in our cells which can control rate of and speed of multiplication of our cells. Usually, if the gene is belong to own cell, it is abbreviated with the prefix C, means cell oncogene, cellular oncogene present in cell. If you talk about viruses which can induce cancers, we have two major groups of viruses. Some can change our own genes, and some viruses can carry their own oncogenes. And if the oncogene is carried by a virus, then it is prefixed with the V letter, means that it is viral gene which is not belonging to our own cell. So what is cancer? Of course, you all can explain what is cancer, and actually we don't have any strict definition in our department because it's not our task to discuss that group of diseases. But actually, I like that WHO definition, and they can explain a lot of things to common people without medical education, and to students it is as well very good. So cancer is a large group of diseases which can start in almost any organ or tissue and manifested by abnormal growth of cells, uncontrolled abnormal growth of cells, usually beyond their usual boundaries. And the main problem with multiple groups of cancers that those cells usually not sitting on one place. Cancer can appear in one organ, but after that, cells will spread to surrounding tissues, to other organs, or even over the body. So according to, again, official report of WHO, in 2018 year, cancer was the second cause of death worldwide, and actually about 10 million people died in that year due to cancer or complications of cancer. Of course, it is a very serious and significant problem which is still not resolved. We don't have any appropriate cure from cancer. We don't have any vaccines to prevent cancer. Of course, we can talk a lot about reasons, about smoking, some radiation, and other, but you all should know it. It's not the task of current lecture. So, how virus can change our cell into abnormal cancer cell? We have several mechanisms which are confirmed by multiple studies. So, first of all, of course, it is disruption of genetic material, disruption of homeostasis when exchange of or some processes in cells are damaged, control is lost, and cells will have abnormal epigenetic integrity. Second mechanism is virus can again interfere with DNA of our cells. It can damage mechanisms which are required to repair DNA integrity, and in general they can instabilize genome of our cell and dysregulate the cycle of cell multiplication or DNA use. Third mechanism is insertion of viral genome into the chromosome or DNA of our own cells. And of course, again, virus can insert some oncogenes into our cell, or it can insert some sequences which will change activity of our genes, and our cell will lose control over multiplication. And again, we have viral oncoproteins that can alter expression of cellular genes and activate inactivate mechanism of apoptosis, or inactivate mechanisms to suppress multiplication of to suppress control over multiplication in our cells. So of course those mechanisms are not full, and a lot of studies are still going on. It's difficult to give some detailed data because studies are difficult, expensive, and of course it is molecular level. It's not available in routine institutions, and only few institutions can perform those experiments. So very often students and even doctors asking, how can you be sure that that virus is really causing the disease? Maybe it is just accidental fighting, it is maybe some just lucky guess or something else. So of course we have several points to confirm or to increase chances that virus is really causing the cancer disease. So if you perform study of tissue or organ affected with the cancer cell, any host cell, any host cancer cell should contain a virus itself or viral genome. Of course, if you have half of the cells without viral genome, it means that those cells are not affected with the virus, but they're still cancer cells. And it is another reason. But if in every cell you will identify viral genome, of course, chance is much higher. So we have some cancers which are present only in humans, some, for example, herpes virus or other. But usually the same cancer should be caused by the same virus in animals. It's prohibited to perform experiments on humans. We cannot infect some human with virus and just wait until that human will develop a cancer. But experiments in animals are allowed, and of course, if you suspect that some virus is oncogenic, you can infect animals, laboratory animals with that virus, and if animals will have the same type of cancer, then of course it is a point that the virus is really oncogenic. So if you perform studies, epidemiological studies on distribution of viral infection, it should correlate or coincide with the rate of cancer. Means that if in some country the same virus of hepatitis B, if in country rate of hepatitis B is much higher than in some other country, then rate of hepatocellular carcinoma should be higher in the same level then in country you comparing with, means if number of common disease caused by virus is higher, then of course number of cancer complications should be higher in appropriate manner. We have markers which shows presence of virus in human body. It's not specific for cancer, but if viral markers are higher in people with cancer, then it is again suspicion for idea that cancer is caused by that virus. Of course, viral infection should precede the cancer. If cancer appeared before viral infection, then the cancer is caused by some other reasons. But if it is always infection, subject is diagnosed with viral infection first, and after some time period, tumor is appearing, then of course again it is a point in viral origin of that tumor. And again, it was that idea, the last point was used in the case of hepatitis B. If you will introduce or invade some measure of control of viral infection, for example, vaccine against hepatitis B, and rate of disease decreased, rate of viral initial disease decreased, then tumor incidence should decrease in the same manner. Means if a common number of ill people will become lower, number of cancers should become lower in appropriate manner. So if all those points are observed in the case of some cancer and viral infection, then it is of course will make ability that disease is caused by virus much higher. If some points are absent, then of course maybe virus tumor have some other origin. So when all those markers were introduced in routine clinical practice, it was actually confirmed that as minimum 7 viruses are causing cancer diseases in human. Of course, maybe in real practice it is much higher number, but for nowadays it is confirmed for only 7 viruses. In general, if you take total amount of human cancers, then about 20% of all cancers are caused by viruses. So 2 viruses already discussed in previous lectures. It is virus of hepatitis B and hepatitis C, which are causing hepatocellular carcinoma, and more than 80% of cases of hepatocellular carcinoma are caused by those viruses. Of course, we have other reasons, but those viruses can cause it for sure. We have human papillomavirus, which is a major virus of all those 7 prevalent in community, and it is the leading cause of cervical cancers and different types of anogenital neoplasms in human population. We will discuss that virus a little later. So herpes virus, such as Epstein-Barr virus and Kaposi sarcoma associated virus, they are confirmed oncogenes. They can cause different types of lymphomas. Kaposi sarcoma virus can cause Kaposi sarcoma, as it is obvious. And tumor-viruses, again, we will discuss today, it is Merkel cell polyoma virus and human T lymphotrophic virus 1 or HTLV1. They can cause as well cancer disorders in human organisms. So till the moment, only 7 viruses approved oncogenes. Of course, we have some suspicious viruses, and if you take some animal viruses, you will find much bigger list of those viruses, but in human body, only 7 viruses officially recognized as cancerogenic.